So today we're going to have a look at genetic disorders. Okay, and the first one we're going to have a look at is Down syndrome. It's one that gets mentioned a lot throughout Key Stage 3 and Key Stage 4. So it's a genetic disorder. It is called trisomy 21. This is because the person with the genetic disorder will have either an extra copy of chromosome 21 or part of an extra copy of chromosome 21. So instead of having 2 copies of chromosome 21, they could end up with 3 copies of chromosome 21 or 2 and a bit copies of chromosome 21. And having those extra bits or extra whole chromosome creates issues, okay, because it doesn't have an extra partner. If you think back to meiosis and the pairing that goes on, if you have a third party, they don't have an extra pair to pair up with, so it can cause some issues. So the parents of the affected child are normally normal, okay, so they won't have the illness. And it's all about the probability of giving birth to a child with Down syndrome. So if you become a mother at 20 years of age, the chance of having a Down syndrome baby is 0.1%. If you are 45 years of age, that massively increases to 3%. Okay? Now, this extra chromosome is thought to be due to complete chance. So there will be no markers in your genome to suggest that you could have give birth to a child with Down syndrome. Just complete chance during meiosis. Okay? So there's lots of screening that goes on during pregnancy. And one is at your 12-week scan. Okay? Well, they will measure the thickness at the back of the neck. And the thicker it is, the higher the probability of Down syndrome. They'll also take a urine sample as well. They'll take into account your age. And then if you are slightly older and other factors have come in, such as the thickness of the back of the neck, they may ask you to come in for an amniocentesis test. So here what they do is they take some amniotic fluid from around the fetus for genetic testing. So they put in a long needle in to the bump, essentially. This will pierce into the amniotic sac, which contains the amniotic fluid, which also contains the baby, and they will draw off a small amount of fluid. This test comes with some risks because obviously you are piercing the sac in which the baby is sat. Okay, so all mothers are offered an amniocentesis test, but those who then have higher probabilities, such as age, such as a test at 12 weeks, will be more encouraged to take them. So if you do have a child with Down syndrome, there's kind of symptoms that they'll have, as well as the facial alterations, or so the facial differences between a normal child and one with Down syndrome will also be a delayed physical growth. They're smaller than normal babies, and they may have mild to moderate intellectual disabilities, so mild to moderate learning disabilities. Okay, so that's Down syndrome. So the next one I want to talk to you about then is haemophilia. Now this is a sex-linked recessive disorder. Okay, so recessive we know as you would have to have, we normally know about them if you think back to GCSE as being like little c, and you have to have the 2 copies of the mutant allele for it to be expressed. Okay, sex linked means then it's on the sex chromosomes. So in this case it's on the X chromosome, and what it is, it's caused by a mutation to a gene found on the X chromosome, and this gene encodes a clotting factor. So factor 8 is the more common version of haemophilia, or factor 9. It's a bleeding disorder. So if you happen to have haemophilia, it could take you longer to clot your blood after you've had a fall or you have cut yourself. Okay, and in extreme situations, this could result in very severe bruising and no clotting at all. Okay, it can affect both males and females. However, because males only have one copy of the X chromosome, if that X chromosome happens to have the mutant gene on it, you are likely to have you're going to have haemophilia. Okay, whereas females have 2 copies of the X chromosome. Okay, and if one of these happens to be a mutant, okay, because you still have a healthy other X chromosome, you will not have the disorder, but you will be a carrier. And actually, females who have 2 mutant X chromosomes generally die when they are fetuses, or they die at birth. Okay, so it's very, very extreme for a female to have 2 mutant copies of the X chromosome. So although males and females can get it, it's generally males that have the symptoms because they only have one X chromosome. Females often are a carrier, which means they don't have it, but if they were then to have children themselves, the likelihood is that their sons will then have haemophilia. Okay, so the treatment for it is literally they give you blood transfusions and replace the factor 8 or factor 9. Okay, and if you have haemophilia in this day and age and you decide to have children, okay, they'll just screen throughout the pregnancy to see whether or not the fetus or the baby has haemophilia, and they'll be treated as soon as they are born. So this used to be a disorder where the people who had it would die quite young, but in this day and age they can live to a ripe old age. So we can carry out a genetic cross to show all of this. So these are the Punnett squares that you will remember from GCSEs and Key Stage 3. So if we have a dad with haemophilia, we'll give him a dot on his X, okay, to show he's got it. And mum doesn't have it, so we give her normal XX. So we're going to carry out our Punnett squares. We'll put mum at the top, dad at the side, put our XX and our XY and put a dot on it and just follow it through. We'll see that they end up with 2 daughters and 2 sons. Both the daughters have 1 mutant X chromosome. Okay, and they will then not have the haemophilia, but they will be carriers for it. So in the next generation, if they have children, the likelihood is some of those children will have haemophilia. And then their 2 sons that they have will not have the disorder. Remember, this is just a probability. Most families don't have 4 children and they don't come out in this order. So there's a chance of a 1 in 2 chance, or 0.5% probability that their daughters, when born, would be carriers. Right, so this is another way of showing it. So haemophilia was made famous in a way by the royal family, okay? And this is a pedigree chart of the royal family. So we can show that males are always drawn as squares, and this is Albert, and circles are females. So this is Victoria, okay? Anyone who's got a coloured in one has got haemophilia, okay? And just by looking at it straight away, we can notice that the ones coloured in are all males, okay? So if this was a question that came up in an exam, we could say straight away that this is sex-linked because no females seem to be affected by it. It's just males. So it must be associated with that X chromosome because boys only have 1 X chromosome. Okay, so we can tell then, using this pedigree chart, okay, that there is an allele in here for haemophilia, okay? We're looking at those that are expressing the phenotype of haemophilia, so the characteristics of it, we're not looking at the genotype of it, so not looking at the precise genes, okay? and it doesn't actually tell us which ones are carriers. We would actually have to work through here. So if this one here happened to have it, but neither Albert or Victoria, we could assume that Victoria was the carrier for it, because if Albert had it, Albert would be coloured in. Okay, and if we look down here then at number 6, his daughter doesn't have it, number 13, but we could assume then she is a carrier for it because her son then has it. Okay, so this is the line that leads down to our Queen. So this is George VI, her dad, George V up here. So this line here shows that none of these had haemophilia. So our Queen's direct descendants did not have haemophilia. Okay, and I've put in this guy here. So this is the Russian royal family. Okay, they were wiped out during the Russian Revolution, but Alexis was very famously known to have haemophilia, and he had quite a severe form of it as well. Okay, so we can tell here that the allele is recessive because it doesn't occur very often. So up here we only see it once out of 1, 2, 3, 4, 5, 6 offspring of Victoria. She had more, but we didn't need to include those. We can see it's only males, so we know it is X-linked, and we could then work through that numbers 2, which is Victoria, 4 over here, okay, 8 because her offspring have got it, okay, 10 because this one's offspring have got it, 12 because Alexis has got it, 13 because their son has got it, and 14 because 2 of her sons have got it. All must be carriers for it. So pedigree charts are really, really useful. Right, so that's haemophilia. So the next one we're going to look at is Huntington's disease. So this is an autosomal dominant disorder. So this means it affects normal genes within the body. So genes that are not found on your X or Y chromosomes. So they're just found on the other chromosomes within the body. And it's a dominant disorder. So if you remember back to capital C, little c. So this was, if you have capital C, you would have Huntington's disease. And so you would be homozygous dominant, or if you had capital C, little c, you'd be heterozygous because you had that capital in there, you would still have the disorder. So a person only needs 1 copy of the defective gene to develop the disorder. Okay, even if your other copy of your chromosome has a non-mutant form, you would still have the disorder. Okay, so it's a disorder which causes parts of the brain to stop working properly over time. Okay, the gene that is mutated causes Huntington's is called Huntington gene, and it is needed by nerve cells in the brain. Okay, so it's needed by the neurons of the brain. When the gene is faulty, the protein which produces repeats, and it repeats certain genetic sequences too many times. This causes damaged neurons in certain parts of the brain. Okay, so the symptoms don't usually develop until you're between 30 and 50 years old. They start showing as difficulty concentrating, stumbling, clumsiness, involuntary jerking, mood swings, and problems swallowing. If you get diagnosed below the age of 21, it's called juvenile Huntington's disease. It's fatal, and usually death will occur between 15 and 20 years after the symptoms start. Okay, so we can do a bit of a genetic cross. So if you are, we'll go with H's this time, capital H, you are homozygous dominant, so you've got 2 mutant copies of the gene. If you're capital H, little h, you are heterozygous. This means 1 copy of the gene, you've got 1 mutant copy, and so you will have the disorder. If you are a little h, so you're homozygous recessive, it means you've got 2 normal copies of the gene, and so you cannot develop the disorder. Okay, this is not a disorder where you are a carrier, unlike haemophilia. You either have it or you do not. Okay, So we could do some genetic crosses here. So we've got a mum here who has a disorder and a dad who does not. So we're going to have a look with mum being homozygous dominant to begin with. Dad is homozygous recessive, and we notice all the children here, okay, will have the disorder. If mum happens to be heterozygous for the disorder, so she still has it, but she's got one mutant copy and one normal copy, dad obviously is homozygous recessive, we notice that 2 children will develop the disorder and 2 children will not develop the disorder. Okay, so if we have a look then, that neither parent has the disorder, so they're both heterozygous recessive, then none of their children will develop the disease. Okay. Right, the final illness we're going to have a look at now is cystic fibrosis. This is an autosomal recessive illness. Okay, so this again, it's not on the X or the Y chromosomes, it's on the normal chromosomes of the body, so not the ones that dictate whether you're male or female, but this time it is recessive. Okay, so you've got to have both mutant copies to have the illness. Okay, so it's caused by a mutation in both copies of the gene, which encodes a CFTR protein. Okay, this protein is involved in the production of sweat, digestive fluid and mucus. If you have one copy of the CFTR, then the person is healthy, but they could be a carrier because their other copy is mutated. There is no known cure for cystic fibrosis. Okay, it's common in Northern Europe, about 1 in 3,000 people have got it, but quite rare in Africans and Asian parts of the population. So the symptoms are an accumulation of a thick, sticky mucus. You have frequent chest infections, coughing, shortness of breath. Okay, it's not only the lungs which are affected, you get thick secretions for the pancreas, so you get then a decrease or a lack of digestive enzymes, so this decrease the absorption of nutrients and can lead to malnutrition, which then results in poor growth. Okay, so if we have a look here then, if you are capital C, so you're homozygous dominant, it means you are normal, you don't have it. If you're little c, it means you're homozygous recessive because they are both copies, okay, are the same, so 2 little c's. This means you've got cystic fibrosis. If you have one normal copy and one mutated copy, because your normal copy is the dominant one, you are normal. Okay, so you don't have cystic fibrosis, but you could be a carrier for it, which means in subsequent generations, your offspring or your grandchildren could have cystic fibrosis. So let's do a genetic cross. So we have a mum with cystic fibrosis, so we give her little c, and a dad who is normal, capital C. If we do a cross, we see that all the children are normal, but they are carriers, so because they are heterozygous. If we then do one where we have got both mum and dad are normal, but they're carriers, so they are heterozygous, we can then see that there's a probability that 1 in 4 of their children will be normal, 2 in 4, so half of their children will be carriers, and there's a probability of 1 in 4 or 25% chance that one of their children will have cystic fibrosis. Okay, so this is a really, really popular topic in the exams. So this is the most recent exam that came out on the BTEC Applied Biology course. Okay, so this came out on the 14th of January 2020. This is a Unit 1 test. Every exam so far I've seen has got something about this topic in. So we can see here, this is all about genetic disorders. So we're looking about a person who inherits 2 identical alleles. Okay, some conditions only affect a person if they inherit 2 identical alleles, so we would say they are recessive. Okay, state the word used to describe with 2 different alleles of the same gene, so this would be heterozygous. So here they're showing you a pedigree chart, okay, and they're asking you about anemia. Now this is not something that comes under this topic, but it's something that would have been addressed when we looked at blood. So anemia is an illness, or sickle cell anemia is very much an illness associated with red blood cells. Okay, so they show you this picture. Okay, so we can see here's dad, here's mum and dad at the top, here's their 3 children, and we can see that this male has got it. Okay, we've got an unknown male here, this male here has got it, this male and this female have got it. Okay, straight away we can notice that not very many of them have got it, so we can probably suggest that it's a recessive allele. Now this was a 9-marker question, okay, and they said evaluate the usefulness of figure 6 when predicting whether sickle cell anemia will affect the unknown male. So they're asking you about whether it will affect this male here. Okay, so the question though is not asking you your knowledge about sickle cell anemia. So writing loads and loads about sickle cell anemia will not get you the marks. The question is asking you all about pedigree charts. Okay, so it's just asking you all about pedigree charts. And if we come back to what we had a look at when we looked at the royal family, this is the sort of stuff that they are asking you for. So if we have a look back then at this, we can see that there is an allele present for sickle cell anemia, so that would get us the mark. We can tell you that this is all about the phenotype of sickle cell anemia, but not the genotype. And it's not told us which ones are carriers. Okay, so these are all things we would write down. So we know we can't tell who are carriers. We can see there's not very many of them, so this must be a recessive disorder. Okay, males and females are being affected, so we could probably suggest that it's not sex linked. Okay, we can assume one of these 2 people here must be the carrier, but we couldn't say definitively which one it is. So for our 9 marker, just by listing all the things that we have just mentioned here, we would pick up those 9 marks. Okay, whereas actually we haven't said much at all or used any knowledge about sickle cell anemia, but we have used all of our knowledge about figure 6, which is the pedigree chart. Okay, so be really, really careful, because it's evaluate the usefulness of figure 6. This could be any disorder they like. Okay, that's not important. This bit here is the important bit when it comes to answering this question. Okay, I would suggest you go away and have a go at this question. It's on the Central Resource Library. So go through. It's a really, really nice paper, so have a look at it. Anyway, I hope you're all well and that this is useful. Bye.